Hello everyone, my name's Steve Carney and I'm the Managing Editor of Drug Discovery Today. I'd like to welcome you to this webinar entitled Genomics Applications Using Acoustic Liquid Handling. Thank you very much to our sponsors at Labcyte for making this event possible. Before we begin, let me remind the audience that we will have a question and answer session following the presentations. Please submit your questions at any time during the presentation using the Ask a Question button on the right-hand corner of your screen. I would encourage you to input questions as and when you think of them. These will be addressed in the Q&A session at the end. The more questions asked, the better the session will be. It's my pleasure to introduce and welcome today's speaker, John Lesnick, Applications Manager at Labcyte. John has worked in the drug discovery and development field for over 20 years. He has contributed assay development expertise for many therapeutic areas, from target validation to preclinical toxicology, and has extensive knowledge in automation and instrumentation. John's expertise with cell biology and biochemical molecular pharmacology has been applied to applications in genomics and drug discovery. So without further hesitation, I'd like to hand over to John to begin his presentation.
Thank you, Dr. Carney, and thank you all for taking the time to join the webinar. We only have an hour, and I want to be sure there is time for discussion, so I want to get started and jump right in to describe some genomic applications using acoustic liquid handling. The content of the webinar is pretty straightforward. To cut to the chase, I'll discuss miniaturization in genomics. This isn't just a topic, but a take-home message, a common thread with acoustic liquid handling that I am hopeful you will see throughout the talk and consider the value in your own research. You will hear about our application roadmap. I'll take some time to describe our approach to demonstrate with context and continuity how you can use Labcyte products in your research. I think it's important because our roadmap provides meaning and purpose to the applications that I will be detailing later in the talk. For me, as an application scientist, I'm not just interested in moving liquid, I'm also interested in the solutions that are created. More specifically on applications, I'll discuss how the EcoLiquid handler can be used for genomic research, including genotyping, synthetic biology, and library preparation for next-gen sequencing. I don't think I'll have time to discuss personalized medicine. I could highlight the work customers have done with the Echo toward personalized medicine, which includes the combination of genomics and drug discovery to help patients. But if this is of interest to you, please contact me and I'll be happy to share this information. Again, I do want to be sure that there is time for questions and discussions on the topics that are covered. So with this slide, I want to point out the benefits and value of miniaturization in genomics. The table lists several applications and demonstrates the cost reduction for each at different levels of reaction volume miniaturization. I will detail later in the webinar qPCR, NGS library prep, and synthetic biology applications. Now, beyond the 100-fold reductions in cost per reaction, the use of acoustic liquid handling offers many additional benefits. This includes using fewer tips and generating less waste, reducing carryover and cross-contamination issues, and can lead to increased productivity as well as throughput. So keep in mind, these benefits are not just for genomic applications, but can be realized in pretty much any area that you're moving liquids, including cancer research, drug discovery, and basic research. Lower reactions use less reagent, resulting in cost savings and often higher throughput. This translates to an increase in productivity without compromising on your data quality. As I mentioned, I think it's important that our roadmap provides purpose to the applications. In reality, our roadmap is a pathway. The PI3K-AKT mTOR pathway. In this pathway, extracellular growth factors binding to transmembrane receptor tyrosine kinases induce PI3K to the plasma membrane. Once there, the P110 subunit catalyzes the phosphorylation of PIP2 to PIP3. PIP3 localizes AKT to the plasma membrane, where it gets phosphorylated by 3-phosphoninositide-dependent kinase 1. After a second phosphorylation event by mTOR complex 2, the fully activated AKT dissociates from the plasma membrane and activates targets that drive increased cell growth, metabolism, survival, and proliferation. The pathway is negatively regulated by tumor suppressor tumor suppressor, phosphatase-intense homolog, which effectively antagonizes the PI3K pathway by dephosphorylating PIP3 to the inactive PIP2. Now, the pathway is at the core of many fundamental biological processes, including RTKs, insulin, and cytokine receptors, which elicit a variety of phenotypic outcomes. There's also involvement with qPCR signal transduction and B-cell receptor linked to immune responses. The aim of using the pathway as our roadmap is to provide a scaffold that is relatable to your research goals. We chose this pathway for the same reason many others, have focused on this pathway. It's well characterized and studied extensively, as you can see by the number of ligands that have been created and drugs that have been developed to intercept or to interact within this pathway. And is a great model system for us to demonstrate the value and utility for using acoustic liquid handling versus traditional methods. From ligands to receptors to enzymes, through signal transduction toward genetic and epigenetic regulation, the controls and tools are available to demonstrate the quality data generated with acoustic liquid handling. Now, the slide could be a webinar in itself to describe the applications toward compound transfer, biochemical and cell-based assays, etc., but that's really for another day. To speak more specifically about the benefits toward drug discovery in genomics, I like to use this cartoon. Not only does it illustrate the pharmacological tools available, but also biomarkers, and points out the genetic variations that can be used to study disease. Now, the knowledge can be applied and translated to new target identification, validation, and additional tools generated, leading to better biomarkers as well as diagnostics. A deeper dive in the genetic variation of two of the proteins in the pathway highlight the importance of genomics in cancer biology. Here we are looking at an old slide showing the percentage of tumors and mutations in either the PI3K-CA or PTEN protein. As you can see, there are many different tissues affected, and in some cases high occurrence rates for both mutations. As a result, cancer is no longer defined by tissue type, but rather by genotype. And at this point, I will introduce a couple of tools we use toward developing applications for real life. One of the tools is the MCF7 cell line. This is a breast cancer line, and it's estrogen receptor positive as well as HER2 positive. The interesting thing about this line, or one of the interesting things, is that it's PIK3CA mutated. And there's an activation in this protein at the point of E545K, which makes it constitutively active. The second tool that we're going to demonstrate and use through the talk is the PC3 cell line. And this is really a prostate cancer cell line. And it is androgen receptor independent, P10 null, and P53 null. Now, one thing that points out with these tools that we have is really that cancer really is pretty tough, and it doesn't just stop with one particular mutation and can actually pile on. So multiple oncogenes can be present within a singular line to drive a variety of cancer types in different tissues. For those not familiar with the pathway or the MCF7 cell line, I have this slide with a couple of cartoons to illustrate the PIK3CA mutations and the mechanism of action in the MCF7s. On the left, you can see the protein broken into specific domains. This includes the P85 and the RAS domains, the helical and the kinase domains as well. The genetic and exon map below shows the prevalence and location of various activating mutations. As you can see, the most common genetic mutations identified include E54K and H1047R mutations. These occur in the kinase and helical domains. The cartoon on the right illustrates what is occurring in the genetically wild-type and mutated proteins. Under wild-type control, the PI3K-CA is in low activity state, bound to P85 at the binding domain and E545 points of contact. In the mutated form, the kinase is only bound to the P85 domain, and the glutamic acid to lysine mutation releases the conformational restraint, resulting in high oncogenic and sustained activity. Similar to the last slide, I provide an illustrated example for the P10 gene and mechanism of action. Starting on the right, this time with the mechanism of action, phosphatase-intense homolog is effectively an antagonist of class 1 phosphatidylinositol 3 kinase, PI3K. In response to extracellular stimuli, insulin growth factors, chemokines, etc., the catalytic subunit of PI3K is recruited to receptor tyrosine kinases, or G protein-coupled receptors, at the membrane through its regulatory subunit, P85 or P101, where it phosphorylates PIP2 to generate PIP3. A lipid phosphatase that antagonizes the action of PI3K by dephosphorylating PIP3 at position D3 to generate PIP2. Now, shown on the left, most mutations affect phosphorylation of the protein affecting activity or the ubiquitination regions affecting recycling. For PC3 cells, P10 is deleted on chromosome 10. The genetic variation in this deletion includes the elimination of many of the exons, and this includes the phosphatase domain. This cell line is effectively P10 null, leading to an unregulated PI3K signaling pathway. To recap before moving on, I spent some time describing the PI3K pathway and its utility and value as a scaffold for our work and some of the tools that will be used, specifically the MCF7 and PC3 cells for this webinar. The aim is to make the experiments more relatable to you and your research goals. Your time is valuable, and my goal is not to simply describe techniques to you. Instead, I want to provide true applications of acoustic liquid handling. Now, that said, let's continue. The first application I'll describe is qPCR. Now, PCR is a common technique, and despite having been used for so long, you can likely still realize value by implementing an echo liquid handler workflow in your lab. The general process for SNP genotyping is illustrated here, where the DNA is typically isolated and purified. Your reagent and sample are added to the appropriate source plates. This is fed to the echo liquid handler to assemble the reactions with low volume additions of costly reagents and precious sample to the PCR plates. This is thermocycled and analyzed, a pretty generic technique. The application we use to demonstrate the benefits of utilizing an echo workflow is for miniaturized PIK3CA mutational analysis. The application note is available on the website and describes the method that reduces sample requirements from 10 to half a nanogram. The assay volume is reduced from 30 microliters to 3 microliters and run in a 384 well format that increases throughput and productivity. This highly efficient and low-cost assay to detect PIK3CA somatic mutations does not compromise the quality of your results. This slide demonstrates the reduction in the sample input. Now, what I used in this particular experiment was control DNA for E54Q mutation. And what I've done is essentially reduce the amount of input that's going to be present in the 3 microliter reaction down from 10 nanograms to half a nanogram. And what we're doing is getting a readout on the light cycler for the VIC channel as well as the FAM channel. Now, a sample is positive for the mutation evaluated, where the VIC CP value is greater than or equal to 25, and the FAM CP value is less than or equal to 38. This is going to be a continued message throughout the next few slides, so it's just a couple of numbers to pay attention to. But as you can see, as we're decreasing the amount of DNA input, we're getting a very nice linear correlation for the amount of CP values present. Here we've got the FAM plotted in the light blue with an R-square of 0.999, as well as a 0.999 for the R-squared in the VIC. The nice thing really is that the slopes are negative 3.74 as well as negative 3.79. This experiment basically demonstrates that we can use less DNA present and still obtain a positive response. For these assays, the limit of detection is important, and the data is shown here. So in this particular case, what I did was take a control DNA, which was E54K positive, and mixed it with different proportions of wild-type DNA, and looked at different percentages of the mutation present. And this serial dilution effectively spanned from 50% mutant or mutated DNA down to 0.5%. We looked at 3 different inputs, at 10 nanogram input, 3 nanogram input, and 1 nanogram input. And again, we have the readouts for both the VIC and the FAM, and we're looking for positive responses. The graph on the right here, we're looking at the fluorescence on the left axis here for VIC, and on the right is FAM, and we're looking at cycle number going across on Y. And what we're seeing is at 1.25% mutant DNA present in a wild-type background, we are seeing positive results of both 3 nanogram input values with a very robust response. Now, proof of concept in control DNA is fine, but again, our aim is to develop true applications. In this experiment, we demonstrate that correct calls can be made in miniaturized reaction volumes and reduce sample input. I isolated DNA from the MCF7 cell line described earlier and ran the reaction at 2 different input values of 10 nanogram input as well as 3 nanogram input in a 3 microliter total reaction volume. In this particular case, a panel of mutant primers were used to detect the presence of a mutation within the DNA tested. And again, the source material was from MCF7s, and we're looking again at the Vic and FAM readouts. And what you can see is that the only positive response, again, which is greater than or equal to 25 for the Vic, as well as less than 38 for the FAM, was for the E54K experiment. And the graph, the amplification plots are shown here on the right, where we're seeing a very nice and robust response for both Vic and the FAM, both at 10 and 3 nanogram DNA input values. Now, correct calls are expected, and what you don't want to see are false positives. To further validate the method, DNA from PC3 cells was isolated. Recall PC3 cells are P10 null, but PIK3CA wild type. Looking again at the miniaturized reaction volume with reduced sample input, the same panel to detect mutants were run. Similar experiment, different input values, different source DNA. And again, this is going to be wild type DNA. And these mutations, or these primer sets here, are going to detect mutations present. And clearly there's no detection of any of the mutations in the DNA sample tested. There's a nice Vic signal, but there's really no FAM, which would indicate the positive nature of the sample for the mutation tested. So to keep things moving along, we can switch gears a little to do some work and look at a little bit of work that was done in synthetic biology. So the workflow here is often repeated to increase size of constructs of a synthetic biology pathway, typically using polymerase cycling assembly. And what you're looking at is the gene synthesis with the echo liquid handler and where it fits in best. So what you can start with are oligos in a 384 well plate and use the PCA reaction. Mix and match any oligo you want, which do come in echo source plates from your vendor. You can run the fusion to join these in specific locations and mix and match pairs. You can run a QC offline and then go on to your cloning. Now, typically, we're going to hand this off to another liquid handler for the transformation, selection, and purification. But when it comes back, you're able to do the NGS library prep for QC or other means of really quality control for your constructs generated. In the last slide, there was a bubble for fusion. And while there are several chemistries available to assemble your fragments in solution, one of the more popular and established is the Gibson assembly method. In this case, a single pot reaction is assembled with the echo liquid handler. Your linearized vector is combined with your engineered DNA fragments, and to this, excuse me, the 5' exonuclease polymerase and ligase is added as a master mix to assemble your DNA. After transformation and selection, you can QC the product by various methods on the echo. Now, as a side note, one of our current projects is to apply this technique to generate various PIK3CA mutations. And this will be accomplished by stitching together in engineered oligos for the helical domain and really step through to identify and recreate different mutational states within the isoform. Stay tuned for those results. Until those technical notes are published, there is a great resource I can provide for miniaturized and efficient Gibson assembly with the echo. Paulina and Patrick Kai's lab at the University of Edinburgh has published smart DNA fabrication using sound waves. This is applying acoustic dispensing technologies to synthetic biology, and you can find this article in PubMed or from our website. So in the study, what they did was essentially step down the reaction volume. Starting on the right here, they're looking at a manual setup reaction, which was 20 microliters, and reduced with the echo the various components of the reaction down to a total reaction volume of 50 nanoliters. What they've done is plot here on the left the reagent costs, and then on the right here on this side is the assembly efficiency in the green. And this is at the various volumes of the concentrations that they have. For the Gibson assembly, they claim that the correct assembly was observed from as low as 250 nanoliter reaction volumes, which was a 20-fold reduction in cost. And we also, quoting them, we also witnessed no background and 100% correct assembly. Now, additionally, the paper describes similar results for the Golden Gate assembly, and they also provide a nice method for miniaturization of PCR in this article. To complement Gibson and Golden Gate, we are writing an application note demonstrating the generation of a recombinant PC3 cell line utilizing the gateway gene assembly chemistry. In this case, we transfected PC3 cells with a synthetic P10 GFP construct. As you recall, the PC3 line is P10 null, and as you can see, the recombinant cells stably express GFP. I should note this is a polyclonal population. And I won't go into all of the details of the method, but briefly, the ECHO was used to combine the gateway reagents with ratios to ensure high efficiency of gene assembly. To this well, the transfection reagent terpofectin was added, and then cells with the ECHO. This reiterates the point that this is a polyclonal population. Now, we don't have time, but this application incorporates several additional applications where the ECHO provides benefits. The first relates to transfection. The reference for optimizing cell line generation with acoustic dispensing is a great resource if you're currently looking at transfecting mammalian cells. As you know, the balance of DNA, transfection reagent, and cells is not a standard recipe. Each of the three variables should be optimized, and the ECHO is a great tool to generate these matrices to optimize the efficiency of your protocol for specific cell lines and potentially a single experiment. The second reference listed is similar with a focus on siRNA transfection. The second application is for cell dispense with the ECHO, and for this I will again direct you to our website for additional details. So as I mentioned, the assembly was run at a ratio to favor efficient P10 GFP fusion, and the destination vector includes a selectable marker for the recombinant mammalian host. There was not a transformation and selection step, so the possibility of a GFP-only cell is not likely, but possible. So to address this, the cell line was sequenced, looking for recombinant P10 in the host DNA. The NGS library prep will be described in the coming slides. So keep in mind that the majority of P10 exons in the phosphatase domain, in particular, are deleted in the wild-type PC3 cells. In this analysis, the sequence is compared to the P10 open reading frame, and we do confirm incorporation of the P10 gene assembly in the recombinant line. So I mentioned in the previous slide that the NGS libraries were prepared with the ECHO liquid handler. The Labcyte liquid handler is capable of accurately dispensing nanoliter amounts of the enzymes and buffers used in library preparation to create miniaturized reactions. This significantly decreases the cost of library preparation per sample and allows reagent to be used for a higher throughput of samples. Additionally, Labcyte ECHO liquid handler can transfer reagents for library QC, miniaturizing reactions such as average fragment analysis and quantitation. Finally, the ECHO liquid handler is capable of both pooling and normalizing samples together in preparation for sequencing. The ability to perform these processes simultaneously gives a workflow time savings unrivaled by any tip-based liquid handler. Here we provide examples of this miniaturization being employed successfully first in the Amyris automated strain engineering group published a miniaturized Nextera workflow in ACS SynBio for plasmid construction. Next, we walk through two of our application notes on miniaturization of Nextera and Nextera XT for human cell, microbial whole genome, and microbiome sequencing. In this application note, we describe a method to streamline and miniaturize Nextera library preparation for NGS. In this note, we use prepared libraries from the PC3P10 GFP cell line and miniaturize the tegmentation reaction 20-fold without sacrificing data quality. The PCR reaction was reduced to 5.5 microliters from the 50 microliter manual method, and by using a 3 to 4 well format, we enable more samples to be prepped by increasing throughput while reducing cost. So the reaction matrix setup included two methods, one that followed the protocol with the Zymo cleanup, and one that used SDS and an additive chemistry workflow to remove the tagmentase. A variety of reaction volumes were run for the tagmentation, including a manual 25 microliter reaction down to an echo-enabled 2.5 microliter reaction, along with various PCR reaction volumes down to 5.5 microliters. The electropharogram from the tape station shows the library prepared from the miniaturized workflow. Prior to sequencing, it is important to get accurate quantitation results prior to normalizing for pooling. The echo is well suited for generating this data in a high throughput manner. On the left is the control curve generated from K562 DNA in the QuantiFLOR1 DS system. The middle panel describes the echo's ability to accurately determine DNA concentration of your libraries at a wide range of concentrations. And these results are fed back to the echo to normalize and pool your libraries very efficiently. The panel on the right is shown to demonstrate that the echo is a non-disruptive transfer. That is, stock DNA, when transferred, is not sheared by the echo. And this slide demonstrates the quality of the libraries generated. The table includes the reactions tested and a variety of quality metrics used to compare the different library preps. As shown, there are no flags when comparing the manual to the miniaturized workflow. The Q30 scores are quite good with no change in the duplication or bias from a PCR reaction, and the diversity of the sample is maintained or is preserved with a percent GC read at 43%, which matches literature values. To further demonstrate the value of the echo for library preparation, we also developed a method for the Nextera XT kit. In this application note, we first show successful miniaturization of Nextera XT workflow for bacterial whole genome sequencing. This miniaturized reaction produces equivalent data to the full-size reaction at a fraction of the cost. Next, we processed a number of bacterial and fungal genomes in a batch using the miniaturized protocol and show that the method is effective across a variety of samples. Lastly, we applied this method to a microbiome study and found that it maintains sample diversity, ratios, and identification ability. So again, the first experiment was to miniaturize the workflow. We reduced the input DNA, the tagmentation volume, as well as the PCR reaction volume. After fragment size analysis, we found that miniaturization added no significant effect to the average fragment generated. The miniaturized workflow did produce equivalent coverage, standard deviation, and Q scores as compared to the large full-size reaction. And this was all done at a fraction of the cost. The electropharogram on the bottom shows the library generated for the 1.25 microliter tagmentation reaction that was amplified in a 2.5 microliter PCR reaction and recovered after a 0.6x magnetic bead cleanup. The next experiment was to look at pooling or multiplexing many samples in a single sequencing run. In this case, each sample was prepared utilizing the 2.5 microliter tagmentation, 5 microliter PCR workflow. The multiplex library sequences aligned well to reference genomes with high confidence. Average fragment size generated during fragmentation was consistent across the diversity of samples and the miniaturized workflow. You will note the sample zymobiomics at the bottom of the table. This sample is a microbial community standard that contains a defined percentage of various organisms. We're using the sample as a control for microbiome work. This is a pool of 10 species that are prepared and sequenced as one, much like what you would do with a patient sample. This leads us to this slide. So Jefferson and I ran the XT studies and did the informatics on all of this. Each sample was aligned to its respective reference genome or close relative from NCBI, and sequencing data were calculated using Biomatter's Genius software and the Genius Aligner. As a result, we see good correlation with the identification and alignment reported and measured GC content. The measured abundance of the standard pool matched well with the expected as a percentage present in the sample. The Labcyte Echo 525 liquid handler enhanced miniaturization of library preparation across a diverse range of organisms, providing consistent, high-quality results for whole genome sequencing despite varying phyla, GC content, genome size, and genome organization. This method can also be applied to microbiome and metagenomic applications as a miniaturized process maintains sample diversity, ratios, and identification. And keep in mind, this work is being conducted routinely by those using the Echo. Elaine wrote a nice paper detailing the method run at Amyris for low-cost, high-throughput sequencing of DNA assemblies using a highly multiplexed Nextera process. And Dr. Chandran points out that any kind of platform that allows us to do the same amount of work in less time at less cost is going to be an integral part of our pipeline. Similarly, there are many organizations currently using the Echo liquid handler for genomic research, including academic, industry, and institute labs. With hundreds more customers worldwide for other applications. With this being a small snapshot of our family of customers. What makes our products stand out is that they create an offering of complementary product lines, including automation, consumables, and software. And at the center is the Echo liquid handler. Echo liquid handler comes in two main varieties. The 525 transfers in 25 nanoliter increments at 5 to 6 microliters per second, and this is ideal for reactions of 0.2 to 10 microliter. The 55X transfers in 2.5 nanoliter increments at half a microliter per second, and is ideal for reactions from 25 nanoliters to 1 microliter. What makes the Echo so cool is that it is a moving liquid with sound. There's no physical contact. It is rapid and flexible, being able to transfer any well to any well, and it's revolutionary. It reduces volume and costs, improves accuracy and precision, eliminates pipette tips, and the potential for cross-contamination. On the right is a cutout cartoon of the transfer process. The destination plate is inverted and drops are shot from the source plate below, and these drops are created as a result of sound energy being focused just below the top of the fluid column. The energy is produced by the transducer, which moves below the source plate. The intelligence of the system is manipulating the transducer to adjust its focus and power relative to the fluid to be transferred, and we call this dynamic fluid analysis. We're able to interrogate the solution in real time to adjust to fluid changes in the source well. By determining the source tension, impedance, and fluid column height, we're able to transfer a wide range of fluid types as well as solutions. This is done without user intervention and is ready out of the box. As a result of the dynamic fluid analysis and the fact that the drop being generated is always the same size, you are able to achieve outstanding precision and accuracy across a wide range of fluid classes. These tables demonstrate essentially what you're targeting as a 50 nanoliter transfer across those solutions that were just described. So in this case, each point between 0 and 60% glycerol is tested looking at a 50 nanoliter transfer, a DMSO at different hydration levels, protein and fetal calf serum solutions, as well as surfactants looking at Triton X from 25 to 200%. Each of the transfer volumes are very close to 50 nanoliters, which demonstrates the high accuracy of the Echo transfer. The bottom table here is describing the percent CVs seen for those same solutions, glycerol, DMSO, fetal calf serum, and Triton 100X, and typically, with the exception of one column there, we're below 3% CVs, which is quite good. Now, another benefit in using the Echo is transferring discrete and small amounts, and this allows you to precisely deliver your reagents. Biopointillism was a term coined from Jeff Buca's lab, where they used the Echo to create some artwork, and essentially here's spotting yeast. And what they've done is generate yeast lines that are going to produce different colors. And from the various sources of those yeast, they're able to place them directly on an agar plate, essentially, to generate this artwork. So in conclusion, I would like to thank you for listening. I am hopeful that the examples of real applications that I presented have stimulated thought to reimagine the way you do science and research to maximize, increase, accelerate, and automate your research efforts. Thank you.
Well, thank you very much, John, for your presentation. I can see that you've all been very busy sending through questions for our speaker. Our first question is just coming up now, and it's the presentation. So, has the echo ever been used for single-cell research?
Thanks, Steve. That's a good question. We're actively involved in that area. It's obviously very popular to be able to isolate single cells and interrogate them as one. So we can approach that from two different aspects. One, at the DNA level, we could do the whole genome amplification in a miniaturized reaction and get very good results in that particular case. And then also there's a fair amount of work being done throughout the world with transcriptome analysis with SmartSeq, either version 2 or version 4. And there are several manuscripts in preparation. So we're hopeful to see those results and data come out in the peer-reviewed journals sooner than later, but they are coming.
Okay. Here's another one coming up. Now, in the example that you showed, was the bead cleanup manual, or were you using robot or automation?
In this case, the bead cleanup is typically done with a liquid handler, tip-based. One issue that we do have with the magnetic beads is their sedimentation factor, where they're falling out of the fluid column to the bottom of the well. So this doesn't work well with the echo where we're transferring from the top. So we work well with a variety of different handlers to let them do that for us, and what we do is take that eluted sample and bring them back for the normalization and pooling with the echo in a single step.
I understand why the echo cannot be used for magnetic bead cleanup steps. Are there other parts of the regular NGS workflow where the echo cannot be used?
For the most part, that's another good one. The bead cleanup is the primary point where we integrate well with others. You will in most cases, you're going to use a DNA prep or purification method offline. This is often a centrifugation or a column purification, where you're going to be isolating your DNA from tissue or other patient samples. My hesitation is the exception there is single-cell work, where we're able to do the whole genome amplification with the echo and then use that DNA directly. So it's not that we can't handle that purification under certain circumstances, but in general, you're going to be using a little bit larger sample, and you'll typically want to be using an offline system to purify and isolate the DNA for the NGS workflow.
Okay. Another question here. It comes asking, you have quite a few workflows mapped out. Do you provide support transitioning from a current workflow using the Labcyte Echo?
Thanks for asking that. That's a fantastic question, because it allows me to highlight just what a great team we have worldwide to support you. There's field application scientists placed throughout the world and in your region that are really excited to work with you and to kind of get into the research that you're doing and to be able to transition it to the echo workflow. There is a bit of a leap. Your destination plates are upside down and you're working with nanoliter instead of microliter volumes. So there's always a little bit of hesitation switching to that. And the field team is great and are more than happy to help you make those transitions.
Okay. Another one here. Recently there's been a question of barcode hopping. It's been quite extensively discussed. Will the echo help with this problem and with newer sequences?
That's a tricky question. The barcode hopping has really come up recently, and my understanding of it is a bit of a contamination issue with the XAMP chemistry for some of the Illumina readers. I believe Illumina has a white paper, and I would point you to that for addressing most of those questions. In terms of the echo and what we can do to help, it's essentially a cleanup question, and the bead cleanups are very good. One thing that we've been thinking about is utilizing some of the enzyme, Exosap, some of the enzymes from ArcEnzyme, say, to help get rid of some of the smaller fragments, barcodes in this case, that remain post-cleanup. So the echo would be very useful there in terms of adding a very small amount of these enzymes to do an additional cleanup post-elution. We haven't proven that out yet, and in-house we don't have that, the exant chemistry reader. So we'd be looking for, a partner to prove that out if people are interested.
Okay, and unless we get some more questions in the meantime, this will be the last question, and I guess it's one that a lot of people wanted to know the answer to while you've been given the presentation, and pretty straightforward. It's how much does the echo cost?
Yeah, that is one of the first questions that come out of people's mouths, and frankly, my answer isn't necessarily a cop-out. As I'm looking through the registrations, we've got a worldwide audience, and that alone kind of it dictates not necessarily different pricing, but different structures within each of the different countries. So I will respectfully defer that to your local salesperson. We have got a global team that are more than happy to talk to you about generating quotes. I wouldn't want to necessarily give you a quote over the phone. So I obviously appreciate the question, but respectfully decline the answers directly.
Yeah, put it onto your salespeople. Yeah. That would be the sensible way for everyone. I'm afraid that that's all the questions that we've received, and I'd like to thank everyone for their participation and for submitting some excellent questions. If there are any other questions that you due to time constraints, you didn't, you weren't able to ask, put them in, and we'll pass them on to the speakers, and they'll contact you later with an answer. I'd like to take this opportunity to thank all the speakers for their presentations and to thank the attendees of today's webinar. It's been a very informative webinar with a lot of thoughtful questions and answers. And don't forget that there'll be a recording of this webinar that will also be available online very shortly. Thanks, everybody, and I hope you have a great day.